When people talk about developing new treatments for rare diseases, the word data comes up a lot, especially when talking about clinical trials and the U.S. Food and Drug Administration, or FDA, review and approvals. You may hear phrases like high-quality data or fit-for-purpose data. But what do those phrases really mean? In simple terms, they describe whether the data used and collected in a clinical trial are reliable enough to help answer two important questions. Does the treatment really help people? And is the treatment safe? When there are problems with clinical trial data, the results become harder to interpret, and it's difficult to clearly understand whether the treatment truly works and is safe. Clinical trials for rare diseases can be complex, in part because they involve small numbers of trial participants. Let's look at some examples where the data from a rare disease trial may have issues, making it less reliable to inform regulatory decision-making. Scenario 1. The data collected is not protected and/or handled carefully. Data must be trustworthy. Before a study begins, there needs to be a clear plan for how data will be collected, saved, and protected. This helps make sure the information stays accurate from the moment it is collected until it is reviewed by the FDA. If the study protocol or the rules of the study aren't followed, even good data can become unreliable. That's because there is no way to be sure the information wasn't changed, lost, or recorded incorrectly. Every step of the data collection process must follow agreed-upon procedures so the results can be verified and trusted. Scenario 2. Not all participants complete the clinical trial. In most studies, it is expected that people may miss a visit or drop out of the study. In studies with more participants, that can often be managed. But when a large amount of data is missing from a small study population, it becomes much harder to interpret the results and understand the true effect of the treatment. With fewer people, there is less information available, making the results less certain. Scenario 3. Treatment and control groups are not comparable. Participants in different groups within a trial must be comparable and assessed in the same way. Sometimes studies compare people receiving a treatment to people who are not in the trial. These are called externally controlled trials. For this type of comparison to work, the treatment and external control groups need to be similar enough that differences between the groups are likely due to the treatment itself. If the groups are too different, it becomes harder to know whether the results are caused by the treatment or by differences between the groups or how outcomes were measured. For example, in the development of the approved drug vutrisiran, which treats polyneuropathy caused by hereditary transthyretin-mediated amyloidosis in adults, researchers were able to use placebo-controlled data from a previous trial for a similar drug. This meant they did not need to enroll another placebo group for the new trial. This was possible in part because the enrollment criteria and endpoints for both the new and the previous trial were similar. If a clinical trial is well designed and the data are of high quality, we can have confidence that the results reflect the true effect of the treatment. When clinical trial results are clear and the treatment has been shown to be safe and effective, much-needed therapies become available to patients. If the results are clear, but the treatment is not shown to be safe or effective, this is also valuable information. These data can help researchers and the disease community better understand the disease and design future clinical trials. Also, when researchers confidently conclude that a treatment is not safe or effective, then time, energy, and resources can be focused on other therapy options and development programs. Developing treatments, especially for rare diseases, is a team effort. Talking with the FDA early in the process of designing a rare disease drug development program can help researchers and patient communities plan studies to collect high-quality and reliable data in the most efficient way possible.